As you all know, gene expression is tightly regulated by transcription factors such as NF-κB, NTF1, SP1, and P53, which all have been shown to be activated in ammonia-treated astrocytes, thereby enhancing the transcription of specific genes. However, most of the genes do not code for proteins, but rather for regulatory RNAs, such as microRNAs, which can degrade mRNA and which can also inhibit the translation of proteins. We already heard about a lot on microRNA expression changes in human brain, in cirrhotics with hepatic encephalopathy, and also in different animal models for acute or chronic liver failure during the workshops here, and there was also a recent publication which focused on that. Besides, many genes are also co-regulated with so-called long intergenic non-coding RNAs, which again can, for example, have an enhanced expression of specific genes. They can inactivate microRNAs, and they can also stabilize the mRNA. In my talk, I want to focus on expression changes of mRNAs and link RNAs, and link RNAs, due to the time limit, only briefly. And please let me first introduce you to the technique which we use. I was asked to do so. So we use a genome microarray, which is quite etablized. And for this, we isolated RNA from post-human mortem brain samples from 8 controls, free from any neurological or liver disease, and from 3 cirrhotic patients without HE, which showed no neurological impairment, and from 8 cirrhotic patients with HE, with showing cognitive disturbances prior to death in coma hepaticum. The samples were taken from the intersection from the parietal lobe and the occipital lobe near to the sulcus parieto-occipitalis. And then RNA was extracted by using standard techniques, and of course they were checked for integrity using a bioanalyzer platform, and you can see here, this is mainly what the system does. It detects the 20S and 18S ribosomal RNA, it produces a quotient from it, and it calculates a so-called RNA integrity number, which is best when it's close to 10, or when it's 10 it's best. And this is an example of RNA which is degraded, and you can see that the RNA integrity number clearly falls down. In our analysis, we included the samples, only included samples which had good RNA quality above a RIN of 7. And those samples and also the groups showed no difference in RNA quality. This array which we use is very robust and works very well with RNA samples which show a RIN of 6 or more. So we next amplified the RNA, and this is simply to illustrate how this works. We are simply producing cDNA out of the RNA using primer which contain a T7 specific promoter. I will come later on to this. And this cDNA is then transcribed into cRNA, and for this we use the RNA polymerase T7, which requires this promoter region. And we use nucleotides which are labeled with the fluorogene Cy3. And then the Cy3 is incorporated in this cRNA. And with this we have a labeling and an amplification process, and we can purify this labeled RNA. And of course, this is also verified that the amplification process and the label process work equal efficient. And in the next step, these samples are hybridized on an array which contains more than 40,000 different transcripts. And then it's detected using a special scanner system. And here you can see an example of 2 samples which show quite different overall intensity. And this is just to illustrate, this is not real value. The real values you can see here, you can see that the overall intensities across the whole array differ slightly. In this case, it's only slightly, but anyway, they are normalized so that the overall intensities, not the intensities of a specific gene, are normalized to the same threshold, and then you can see the intensities are very comparable. So next, what we do is we translate the reporters which are expressed on the microarray into genes, and we identify genes which are upregulated and downregulated in a discriminatory gene analysis. And those genes which show differential expression are then put into a functional grouping analysis and therefore we employ databases, and then they can be annotated to biological processes and functions. And this is what you get from this discriminatory gene expression analysis. This is a heat map showing you the relative expression levels of the different genes. And you can see every sample which is included in the study, and you can see some genes which are color-coded in red. This indicates an upregulation of those genes, or a high expression level, as you can see here. And you can also see that some genes have a green color, and this is indicated for rather low expression level. So when we compared which transcripts show a differential expression as compared to the controls, we found that about 1% of all transcripts which are represented on this array are changed in the brain of patients with liver cirrhosis without HE, whereas liver cirrhosis patients with HE have a change in about 1.5%. When we compared all those genes which show a differential expression in the group of the patients with in the cirrhotic patients without HE and with HE, then we found that only a small subset, there's only an overlap of a small subset of those genes. And from those 22 genes which are regulated under both conditions, only 6 of them are concordantly regulated, which indicates that you have specific changes in the transcriptome in the brain in cirrhotic patients which depend on the presence of hepatic encephalopathy. We next searched for biological processes or for an enrichment of those genes in certain biological processes, just to show which pathophysiological mechanisms might be involved. And as you can see here, we found an enrichment of those genes with different expression level in the brain of liver cirrhotic patients with hepatic encephalopathy in many categories which have recently been postulated to play a role in hepatic encephalopathy, such as inflammation or receptor signaling. And when we analyzed the functions which these genes are involved in, we could show, or we found that several of those genes are involved in stress response pathways, and especially in oxidative stress response pathways. And here are some of them shown. And this is in line with recent data which came from our lab, where we could show that there is indeed an increased or evidence for increased markers of oxidative stress in the brain of patients with liver cirrhosis and hepatic encephalopathy, but not without hepatic encephalopathy. And I just want to mention some of them, because, for example, the hemoxygenase 1 gene is often referred to as a surrogate marker for oxidative stress, and it has been found in many different experimental models for hepatic encephalopathy, such as ammonia-treated West astrocytes, and in different in vivo models for acute and chronic liver failure, which is surely a marker for oxidative stress, but what is often overlooked, it has an anti-inflammatory effect, because it may inhibit the degradation of the I-kappa B alpha, and thereby terminating NF-kappa B inflammatory responses in the brain. What is very interesting is that we also found an upregulation of genes which are part of the antioxidative stress response, such as peroxyredoxine 4 or selenoprotein S. They can detoxify free radicals, such as hydrogen peroxide and probably also peroxynitride, and they serve therefore as a very important function. We also found that some transcription factors which are involved in the expression of genes which have antioxidative properties are upregulated, like PPAR alpha. Yesterday, Markus Jörgens showed that in ammonia-treated astrocytes, MRP4 is upregulated in a PPAR alpha dependent manner, but PPAR alpha also can interfere with NF-kappa B signaling and also interfere by, for example, with the upregulation of inducible nitric oxide synthase. In recent work, we could also show that hyposmotic astrocyte swelling and ammonia leads to the liberation of zinc ions from zinc sulfur clusters, and this leads then to the activation of transcription factors with high affinity towards zinc, such as metal responsive transcription factor 1. And this is, leads then to the upregulation of metallothioneins in cultured cells. This is very important because zinc is very toxic to the cells, so it has to be sequestered. And this data is now corroborated by our gene chip error, where we can show that in the brain of patients with liver cirrhosis and hepatic encephalopathy, there's an upregulation of many of those metallothioneins. And interestingly, the expression level of those genes in those patients correlate with the serum ammonia levels. And this is true not only for this metallothionein, but also for several others. Of course, we were also interested in looking at genes which are involved in inflammation, because this is a very interesting topic now in hepatic encephalopathy. And we could recently show that microglia becomes activated in the brain of patients with liver cirrhosis and hepatic encephalopathy. And again, with this gene chip, we could show that there is an upregulation of many genes which are clearly indicative for activated microglia. And this is also has recently been confirmed by Claude Dennis from the University of Sydney, who showed beautifully by immunohistochemical analysis that in the brain of patients with liver cirrhosis and HE, microglia adopt a phenotype which is characteristic for an activated microglia. But interestingly, we also found that there's an upregulation of many genes in patients with liver cirrhosis and HE, which may suggest that those microglia adopt a so-called M2 phenotype, which is an anti-inflammatory phenotype. Moreover, we were looking at very prominent genes which are involved in inflammation, like toll-like receptors on the interleukin-1 receptor, for example. And from those, it is known that they also activate, through activation of the NF-kappa B signaling pathway, the transcription of pro-inflammatory cytokines. And indeed, we found an upregulation of many of those genes of toll-like receptors, toll-like receptor 1 and 2, which is specifically expressed in the microglia in the brain, in the patients with liver cirrhosis and hepatic encephalopathy, but not without hepatic encephalopathy. But more interestingly, we found also that several genes are upregulated, such as IRAK4, for example, which have anti-inflammatory properties. They can block the signaling from toll-like receptors and also from the interleukin-1 receptor. This could be part of a counteracting mechanism terminating inflammation. We found also that some of those genes were also correlated with the expression level with the serum expression levels of C-reactive protein, which might indicate that systemic inflammation might have an impact on cerebral gene expression. In order to verify this data, we took samples which were derived from Australia, and whereas the European cohort, which was analyzed, was from this region, the sample from the Australian cohort was taken from the Gyros fusiformis. And we repeated the array with those samples, and again we could clearly show that we have an upregulation in both cohorts of many oxidative stress markers, such as hemoxygenase 1 and those metallothioneins, which I recently introduced, and also in microglia activation, and also markers for inflammation. So let me briefly or shortly come to the long intergenic non-coding RNAs. We also analyzed the expression level of such regulatory RNAs in those patients using microarray technique, and we analyzed about, this array comprises about 8,000 micro-link RNAs. You can see here that in the brain of the patients with liver cirrhosis without HE, we have a downregulation of many of those links, but also an upregulation. And the same is true for the patients with liver cirrhosis with HE. However, again, if we compare those genes with altered expression level, we can see that indeed there's only a very, very small overlap. Again, showing you that you have specific expression changes of genes in liver cirrhotic patients with HE. So let me summarize what I've shown to you. I've shown to you that we have specific transcriptome changes in human brain in liver cirrhosis that depends on the presence of hepatic encephalopathy. And those changes involve genes which are involved in oxidative stress, microglia activation, and anti-inflammatory pathways. And that peripheral inflammation may trigger cerebral gene expression changes in a chief. And with this, I want to thank all the people who were involved in this study. First of all, my mentor, Professor Dr. Dieter Heusinger, our technical assistant, Thorsten Jansen, Dr. Hans-Jürgen Bittmann, and all the people from the Australian Brain Donor Program, which helped us a lot and which were very helpful, and the people from Miltenyi Biotech, which supported us in performing this array. Thank you very much for your attention.
Thank you.